Okay, so where do we stop? We stopped. So now we're back to the slides. 2.3, the mixtures. Okay. Okay. So we mentioned that energy is a form of power that gives us the capacity to do the work. So to do the work means to move something, such as a muscle or a molecule. So there are different types of energy. There is potential energy, and that's the energy stored in an object, but not currently doing the work. So an example would be a water behind the dam that has potential to spill and move the rotors of and move the blades of a solenoid, and in the middle of it there would be a copper iron with magnet to create an electric field. But that's potential energy. Same thing with the chemical energy. The chemical energy, I have ATP. ATP is adenine nucleotide with 3 phosphate groups. That's why we call it adenine triphosphate. Adenine triphosphate. If I break one bond, it's going to now turn from adenine triphosphate into adenine diphosphate. Adenine diphosphate. So was that catabolism or anabolism? Catabolism. And catabolism liberate energy. Give me energy. So now I'm going to get energy out of this catabolism. I'm going to get energy out of this catabolism. So we're going to see that also in the next slides. So the potential energy is the energy stored in an object, but not currently doing work. An example would be the water behind a dam. And there would also be chemical energy. That is the chemical energy in the molecule, like the ATP. Okay? And there's also free energy, and that is the potential energy available in a system to do the work. Then there's kinetic energy, and that's the energy we use to power osmosis. So when we had on one side 500 millimoles of salt, and on the other side we had only 100, what made the salt move from an area of a high concentration to an area of a low concentration? It's the kinetic energy, the bumping, the movement of these molecules start bumping to each other and then getting kinetic energy and moving to another from one place to the other. Okay, kinetic energy is the energy of motion. So we see that is what powers the motion of molecules from one place to the next, and energy doing the work. So examples would be muscle movement, the flow of ions, and the vibration of the eardrum. Heat is kinetic energy of the molecular motion. So now we get the molecules moving. So if you look at water, when you heat the water, so you get it first as ice, it's very cold. When you heat it, it's going to melt. Now it's going to become water. So the water molecules are moving more than ice, solid ice. Liquid water is moving faster than solid ice. Then when you heat liquid water, then it's going to become the gas. Then it's going to move faster than the liquid water. So that shows you temperature is the average velocity of molecules. So the hotter things are, the more movement there would be. So heat is the kinetic energy of molecular motion. Electromagnetic energy is an energy where we have the movement of the photons, the electrons. So electric energy is the movement of electrons. Electromagnetic energy is the movement of photons. Photons are emitted when an electron moves, when an electron falls from an area of a high energy to an area of a lower energy, it will emit a photon. And so now you're just appreciating the different types of energy. You don't need to understand, like I'm not going to ask you what is a photon and how does the photon emit the energy. But for you, it'll make you appreciate that we have heat energy, and then we have kinetic energy, and then we have potential energy. Just know the big ones, but you don't have to know, for example, the details. the difference between an electromagnetic energy and the use of a photon. And when we were doing the acids and bases, negative the logarithm of hydrogen concentration. One of the students was asking me, what is the logarithm? You just don't need to know that. Just understand that it is negative the log of the hydrogen concentration. Because if there's a question on the exam, it's going to be that the pH is negative log of OH concentration. That's wrong. pH is negative log of the hydrogen concentration. That's right. That's all you need to know. But you will not be asked, what is the logarithm? You will not be asked to calculate the logarithm. This is not calculus. Okay. Now we're going into the chemical reaction. So the chemical reaction is like a transaction. You go to the store, you give the money, you buy something. Now, a chemical reaction is a process in which a covalent or ionic bond is formed or broken. So, in a chemical reaction, we form a bond or we break a bond. So, always the arrow in a chemical reaction is always pointing from the reactants to the products. And if products are falling back to give me a reactant, then they become the reactant. Okay? Then they become the reactant. So if you heat up liquid and liquid becomes gas, then the reactant is liquid water and the product is gas. But if you now decrease the temperature and then the gas is starting to fall back into liquid, now what used to be the product is now the reactant is coming back to become a product. So now let us now look into the different classes. That one you need to know. The difference of the chemical, the difference between the chemical, reaction. We have the first type of chemical reaction. We have the decomposition, where you have A and B together, and they decompose into A alone and B alone, like NaCl decomposing into Na plus Cl minus. That's a decomposition because it decomposed. Decomposition reaction. What is the second type? Is the opposite. When you have two separate reactants like Na plus and Cl minus, and they both add up together to give you something big of them together joining, and that would be NaCl. Then we have something where we do not have big fall. So this is an example of catabolism. That's something broke down. And this is an example of anabolism. Things are building up. There's another example where we have a third type. So this is decomposition. This is composition reaction or condensation reaction or composition reaction, decomposition or decondensation, decondensation reaction. So the names from books different, but the idea is the same. Then we have a second type where you would have A and B together react with C and D, and the product would be, now A will no longer be with B, so you would have AC, and you end up with, so now you have AC and BD, So now they exchange, there's an exchange. So that's an example of a displacement or exchange reaction. That's an example of exchange reaction. Okay? That's an example of an exchange reaction. So let's look. That's an example of a decomposition reaction where you had large molecule breaking into macromolecule, like going from maltose into two glucoses, going from a disaccharide into two monosaccharides, going from a polymer into a disaccharide into two monosaccharides, going from a big polypeptide chain into little amino acids, going from lipid into glycerol and free fatty acids. Then there is synthesis reactions or composition reactions where two things build up. Now you go the other way around. Two glucoses come together to give you maltose. Maltose joining a polymer to give you a sugar polymer. Then there's exchange reactions. Exchange reactions when things alternate, like what's happening between when there is a reaction between stomach acid, HCl, ACl, and bicarbonate, NaHCO, HCO3. What happens is the H is going to be picked up by the HCO3 because that's a buffer. So now we will have H2CO3 plus, and then the sodium is gonna, and then the chloride is gonna join the sodium because that's HCl. The chloride is gonna join the sodium to give us NaCl. That's a salt. That's a salt. So that shows you we can do replacements. So now you're just appreciating the different types of reactions that can happen. So what is that an example of? When you have a starch molecule that is broken down into little glucose molecules. That's an example of decomposition and catabolism. Here, amino acids, little amino acids coming together to give me a big protein polypeptide chain, a protein molecule. That's an example of a synthesis reaction. Then we have an exchange reaction. What happens with the exchange reaction? In the exchange reaction, you have A and B and C and D coming together to give you AB. So they will alternate. You had A and B, C and D, then they would all join together. Then they would split by A no longer with B. A is now with C, and C no longer with D. D with B. Okay, reversible reactions. So reversible reactions can proceed in either direction under different circumstances. So it depends on where the stress is. So look at this example. You have CO2 plus water will give you carbonic acid. If there is a lot of carbon dioxide, the reaction is going to push this way. We're going to make a lot of carbonic acid. But if there is a lot of hydrogen proton, the reaction is going to move this way. Then we're going to start to absorb the hydrogen proton and to make bicarbonate. So this is carbonic acid and this is bicarbonate. If there is a lot of acidic carbon dioxide, then we're going to be forming carbonic acid. But if there is a lot of hydrogen proton, we're going to be forming bicarbonate. So this is an important reaction in the respiratory, urinary, and digestive physiology. We're going to learn about that in the future, and that is how the buffer system. So the one in the middle, carbonic acid, that's a buffer. So when there is a lot of hydrogen proton, it will take them. But when there is a little bit of hydrogen proton, it will donate hydrogen protons to drop the pH. So there are reactions that are reversible. They can go in both directions depending on the stress. This concept is really important. If you put a lot of stress on one side, the reaction is going to go on to the other side. If you put a lot of reactants, the reactants are going to be pushed to go into the product. But if you put a lot of product, the product is going to flow back into the reactant. So the reaction will go in the direction of most stress, from an area of a high stress to an area of a low stress. Does that make sense? So if there is carbon dioxide, so the stress is represented by here. So if there is a lot of carbon dioxide in the blood, the reaction is going to go in that direction. So we're going to be absorbing a lot of hydrogen proton, leading to carbonic acid. In the other way, if there is not a lot of carbon dioxide, there is a lot of hydrogen proton, we're going to be going in the opposite direction. We're going to be absorbing this hydrogen proton. So we always obey. When there is stress, we go the other way around. That makes the stress go down. Are we clear? Okay. Reaction rates, how fast the reaction go. What causes the reaction? What causes the reaction to go faster? Number one, the concentration. We talked about that. Remember when I said if there is a lot of one reactant on one side, it's gonna push the reaction on the other side. That's one of the reasons that's gonna speed up the reactant, and that is the concentration. Number two, the temperature. If you heat up the reaction is going to go a lot faster. And then you have the catalyst. A catalyst is an enzyme. This is really important. So if we have an enzyme, and the enzyme, this is the time. Time goes on. And this is the concentration. You have reactants. Reactants that you need to make them into products. So this reaction must go up a route and then to give you, so the reactants have to go uphill. They have to go uphill in order to go from reactants to products. And this uphill barrier that they have to cross is known as EA, or activation energy barrier. EA, activation energy barrier. So in order for reactants to go to products everywhere in the world, wherever there is a reaction, we go uphill, up an activation energy barrier, we cross that. So what do the enzymes do? The enzymes, they lower the activation energy barrier and tell the reactants, listen, you don't gotta go route A, that's too far. I have an alternative shorter route. Let's go this route. So now they really have dropped the activation energy barrier. So they lower the activation energy barrier that must be surmounted in order to go from reactant to product. Now, the problem is many students, they think that the enzyme is, when it does that, it's going to disappear. So they think that the enzyme, when it pushes the reactant to product, then no, the enzyme is still going to be there. It doesn't get consumed in the reaction. This is one of the properties of the enzyme. The properties of the enzyme is that the enzyme, number one, the enzyme does not get consumed in the reaction. It is not part of the reaction. It just makes it go faster. Number two, the enzyme does not change the reaction. So if the reaction says we have AB plus AC, and we're supposed to end up with AB plus CD, plus CD, we must end up with AC plus BD. The enzyme is not going to say, no, let's keep it AB, and or maybe ABD, and then you leave the C alone. No, it's not going to change anything. The enzyme is not going to change the reaction. The reaction is going to be the same. The enzyme is not going to get consumed in the reaction. It's going to still be there. It's ready to do some more as long as you give it the ATP. So these 2 points are really important. We're going to see now. Enzyme catalyst. So now if you see in the slide, the enzyme catalysts bind to reactants and hold them in orientations that facilitate the reaction faster. And catalysts are not changed by the reaction and can repeat the process frequently. They are not changed and they're not consumed. Now we move on to metabolism. Metabolism, we alluded to that. Metabolism is divided into catabolism and anabolism. And they are not enemies. They work together. The energy we get from catabolism, we use it to build metabolism. And we build metabolism, so later when we need catabolism, we use catabolism. So they work together. Anabolism and catabolism, they participate together. So in metabolism, all the chemical reactions of the body, metabolism represents all the chemical reactions. So every reaction in your body is either part of catabolism or part of anabolism. So catabolism. Catabolism is an energy releasing decomposition reaction. Now they are introducing a new word, exergonic. Exergonic. What is exergonic and what is endergonic? So these are words, you know, sometimes in anatomy and physiology, we have our own terminology. So you already know that catabolism, you know that catabolism means breakdown. Breakdown. And breakdown means we get plus energy. We gain energy. We gain energy. We gain plus energy. Now, the word they use for that is exergonic. Exergonic. Why exergonic? Because the energy exit. So if I have a chain of sugar or a chain of fats or a chain of proteins and I break it down, energy is going to get out of that. We say this with the ATP. When we broke down the ATP phosphate group, the 3 phosphate groups, when we broke one of them, they're attached to adenine. When we broke down one, what happens? We convert it from ATP, adenine triphosphate, 3 phosphate, into adenine diphosphate. Very good. To adenine diphosphate. And when we went to adenine diphosphate, we got plus energy because we broke the bond, so the energy was liberated, exited. That's why they call it exergonic, because the energy exited. So that's an exergonic reaction. So catabolism, does it take energy or does it give off energy when you break down? So does the energy exit or does the energy enter? So is that an exergonic reaction or an endergonic reaction? Exergonic. Excellent. Okay, let's say now we move to anabolism. So now we know that anabolism means build up. So now we have little monosaccharides or free fatty acids or amino acids. Now we're going to join them. If we're going to join them, then we're going to have to take energy to do that. So we're going to consume energy. So energy is going to enter into making these bonds. And when energy enters into making these bonds, that is known as endergonic reaction. So you must know that catabolism means breakdown, means give us plus energy, that's exergonic. Anabolism means build up, means endergonic, means we consume energy. Clear? Any questions? Okay. So now let's look. Catabolism is energy releasing, exergonic decomposition of the reactions, and it breaks the covalent bonds and produces smaller molecules. Let's look at anabolism. Anabolism is energy storing, endergonic, energy storing, energy consuming synthesis reaction, and requires energy input. You see, it requires energy, it's energy consuming, requires energy input. Like, for example, the production of fat. You see, production of fat. So that's anabolism. In catabolism, it wasn't production, it was smaller. We don't make, fats are big. So you need to know that. You need to know that fats are macromolecules. They are made up of little micromolecules. What are the micromolecules that make up fats? Free fatty acids and glycerol. Excellent. What are the macromolecules that make up proteins? Amino acids. Excellent. What are the micromolecules that make up the sugar polymer? Monosaccharide, not just glucose. Remember, there is fructose, there is galactose. So there is monosaccharides. Together, they give us disaccharides. Together, they give us polysaccharides. Who are the 3 disaccharides? The disaccharides. Lactose, the milk sugar, and maltose, the sugar of the oatmeal, and sucrose, the sugar of the sugar cane. Now, sucrose is made up of what? That's the oatmeal. So the easiest one for you to remember is the maltose, the oatmeal sugar. It has double glucose. That's the easiest one. Which disaccharide has double glucose? That's maltose. Now what you just need to remember is lactose, and lactose has galactose in it. So that's out of the way too. You know that all of them have glucose. All of them are disaccharide, and of the two, there's glucose always. You already ruled out maltose because it has two double glucoses. So that's one out of the way. Galactose makes up lactose. No, galactose doesn't have lactose in it. Lactose has galactose in it. Yeah. So glucose and galactose, and then fructose and glucose together, they make up sucrose. Okay? Yeah, you got to know that one. Okay. All right. Moving on. So this breaking down of bonds, these buildup of bonds is taking place through oxidation and reduction. Oxidation and reduction. So oxidation, Leo says, Ger. If you lose electrons, you're oxidized. If you gain electrons, you are reduced. So in sodium chloride, who gained the electron? Chloride. So if you gain an electron, you are reduced. If you lose an electron, you are oxidized. Now we got to know, if you are oxidized, you are the reducing agent. And if you are reduced, you are the oxidizing agent. So it's pretty simple. If you are oxidized, then you are the reducing agent. You are the agent of the opposite of what happened. If you are oxidized, like chloride, like sodium, so sodium lost an electron. That's why it's positive. So Leo says, Ger, lose electron oxidation. If sodium lost an electron, is oxidized, what agent is it? It's the reducing agent. You cannot be oxidized and you are the oxidizing agent. No, you are oxidized, you are the reducing agent. If you are reduced, you are the oxidizing agent. So in sodium chloride, Na plus lost an electron, it is oxidized. And if it's oxidized, what agent is it? Reducing agent. And chloride gained an electron. If chloride gained an electron, that makes it reduced. And if it's reduced, what kind of agent is that? Oxidizing agent. So, oxidation, a chemical reaction in which a molecule gives up electrons and releases energy. And a molecule is oxidized when it loses electrons, and the oxidizing agent is the electron acceptor. Okay, reduction, any chemical reaction in which a molecule gains electrons and energy. So a molecule is reduced when it accepts electrons. The reducing agent is the molecule that donates electrons. So the oxidation of one molecule is always accompanied by the reduction of another, known as oxidation, reduction, redox reactions. We said that organic compounds have what in them that makes them organic? Carbon. And inorganic compounds have everything in the world except carbon. Very good. Okay. Carbon compounds and functional groups. These functional groups, you may not need to memorize each single structure, but you have to know the functional group. You have to know hydroxyl. That's OH. That one you got to know. OH. Hydroxyl is OH. Alcohol is OH. Amino, that's NH3. We're going to see that in a little bit. So we talked already about that. We gave already a very good introduction about our main 4 macromolecules. You can either be a carbohydrate, you can be a lipid, you can be a protein, and you can be a nucleic acid. Nucleic acids are only in the DNA. We do not use them as a food source, but we use them like in ATP. That's a nucleotide. We use it to give us ATP. How? By removing one of the phosphates and converting from ATP into ADP, and that is an example of a endergonic or an exergonic reaction. Exergonic. Good, good, good. Very good. Very good. My voice is not going away for nothing. I'm good. If one walks out with a wealth of knowledge, I am so happy. Okay. So now the carbon compounds. The carbon compounds and functional groups. These are the functional groups that we got to know. The hydroxyl, the methyl. If you pay attention really well in this course, you will take the NCLEX like that and you will pass it before anybody else. Just be good businessmen, businesswomen, and invest well so you don't have to do the job twice. Investing really deeply in this course is really going to help you a lot and going to help you with the other courses too, microbiology, with pharmacology, with pathology, because all this is the backbone. This is the building block for all of them. Okay? Okay, so the carbon uniquely suited to form a variety of structures. Number one, carbon, this organic compound. It has 4 valence electrons. I'm not going to ask you how many valence electrons are there. So that's general information. Can form 4 covalent bonds. We've seen that in the amino acid. Is it a secret that carbon has 4 valence electrons and it can form 4 bonds? We saw that in the amino acid. With which carbon was the name of the middle carbon was called what? The middle carbon, I drew it for you and I put a sign in there. The art? No, the R is one of the 4. Remember I said north, east, south, west. So these are 4 bonds. And the carbon is in the middle. What's the name of the carbon in the middle called? And I put this sign. I said like this. You have H at the top. You have COH on the east. You have NH3 on the west. You have R in the bottom. And you have carbon in the middle. No. That's the alpha carbon. The alpha carbon. It's the one in the middle. It's the alpha carbon. Okay. It can bind readily with other carbon atoms to form carbon backbones. These are long chains, branched molecules, and rings. So readily bonds with hydrogen, oxygen, nitrogen, sulfur, and other elements. So the carbon backbones carry a variety of functional groups. These are what you call hydroxyl, methyl, carboxyl, amino, phosphate. These are the functional groups. These are the R. These are the R. So in the R, I can have a OH, I can have a methyl, okay? I can have valine has a methyl group in where the R is. Something really special about the amino acid is that there's one amino acid. That one, only one. That one, you got to know it. It has double H's. So its R group is also an H. So it has one H and the R is an H. That's glycine. That's glycine. So on the exam, which amino acid has an H terminal, an amino terminal, a carboxylic terminal, and an R terminal that happens to be another copy of a hydrogen? What's the name of that amino acid? Alpha carbon. The name of the amino, I'm looking for the name of the amino acid. It's known as glycine. That's the amino acid. Amino acid. Okay, glycine. Okay. Okay. These are the carbon backbone carry a variety of functional groups. These are attached to the carbon backbone. So hydroxyl, methyl, carboxyl, amino, and phosphate. We're going to look at that. So look at that. That's them. These are the functional groups. You need to orient yourself with them. So OH, that's the hydroxyl. OH is a hydroxyl. And where do we find the hydroxyl? In alcohol and sugars. Sugars have a lot of hydroxyl, and alcohol have a lot of hydroxyl. That's why we get a lot of reactive oxidative species. When we were talking earlier today about free radicals and reactive oxidative species, you drink the alcohol, it has hydroxyl, then it will metabolize into acetaldehyde, and the O is going to turn into sucrose. Alcohol is going to turn into sucrose. The oxygen, the OH of alcohol, is going to turn into a free radical. Free radical with electron minus is going to turn into O minus 2. O minus 2. O minus 2. We said we use the dismutase to repair that. The antioxidant, earlier today we said we take our antioxidant and there is the enzyme SOD superoxide dismutase. So this OH is going to turn into what? Yeah, so that's the superoxide dismutase. Repair it, excellent. It repairs it by turning it into an oxygen we can use for oxidative phosphorylation, for breathing, for aerobic respiration, and also it's going to turn it into hydrogen peroxide. But what is the bad thing that we are repairing called? Somebody says sucrose. It's not sucrose. What is the name? It is O2 minus. We established that. That's why they call it a free radical. Why they call it a free radical? Because it has a minus. If it's O2, that's what we want. If it's O2 like this, that's what we want. But it's not O2. It's O2 minus. There's 2 electrons there. So we need to get rid of these 2 electrons with the help of SOD, superoxide dismutase. What's the name of this dangerous thing? Superoxide. That's why they call it superoxide dismutase. So you see how the hydroxyl can turn into a superoxide to destroy us. So the functional group is OH. Another functional group is CH3. CH3, that's methane. You can find a lot of that in the fats, in the oils, in the amino acids. You're going to see it in a little bit. You're going to see a lot of CH3. Then there is the carboxyl, where you have OH but with another CO. That's a carboxyl. A carboxyl group looks like that. Double bond with a C, then an OH. Okay, so you have an OH with another CO. That's a carboxyl. So this is just familiarizing yourself with them. Now that's the amino. You need to know the carboxylic terminal because that's one of the sides of the amino acid. So one of the sides, north, south, east, west, one of the sides is a carboxylic side. And the other side is the amino side. Then you have a hydrogen side, you need to know that. And then the last one is the R group. The R group could be anything. It could be a carboxylic, it could be a methyl, it could be a hydrogen, it could be a phosphate, it could be anything. Okay. Macromolecules are large organic molecules with high molecular weights. So most macromolecules are polymers. And molecules made of repetitive series of identical or similar subunits known as monomers. So we know that the monomers give us polymers. Monomers may be identical or different. So examples are starch is a polymer, about 3,000 identical glucose monomers. That's a starch. So starch is like a lot of glucoses attached together, like a train. You know train cars attached together? Okay. So DNA is a polymer of 4 different nucleotide monomers. So polymers formed by polymerization, which is the joining of the monomers. The joining of the monomers. Monomers and polymers. There is polymerization. So monomers covalently link together by dehydration. We talked about that. So if we want to join things together, you remove water. Dehydration, synthesis, condensation. You condense them together. Then a hydroxyl group, OH, is removed from one monomer and a hydrogen group from another, and water is produced as a byproduct. Then there's hydrolysis. Hydrolysis is the opposite of Dehydration synthesis. Hydrolysis is the opposite of dehydration. So in hydrolysis, we add water to split the monomers. And enzymes help break the covalent bond that links two monomers together. So a water molecule ionizes into OH minus and H plus. And OH minus is added to one monomer and H plus is added to the other monomer. So here we see these are two monomers. So do you get it now? Do you see how we have dehydration? You see now we have dehydration and hydrolysis. Look at what happened in dehydration. So that's H2O. That looks like water. So one side will donate the OH while the other side will donate the H. And then water will be taken out. And then both of them will be connected like a dimer. You see the dimer? So water has been removed because one H from here, OH from here, OH plus H, that's H2O. Then a bond is made, and that's how we make dehydration synthesis. We synthesize by removing water. This one is really important. Which of the products, which of the actions are mediated in dehydration synthesis? It is the removal of water. The removal of water gives us the dimerization. But in hydrolysis, we split, so we add water. So you need to know when we add water, when we remove water. We add water when we want to split. You see, right here, we added water. You see the arrow? The arrow is pointing in. So we added water and we split blue from orange. We added water to split blue from orange. We got two monomers. And here, we removed water. We removed water in order to, we removed water, and that led to the joining. So you see the arrow pointing out. See the arrow pointing out? So if the arrow pointing out, that's dehydration. We lost water. That's minus water. We removed water. When we remove the water, we condense. They stick together. And here the arrow pointing in. So when the water goes in, we split the dimer. So we do hydrolysis to split, and we do dehydration to join together. That's very important. Okay, then we move on to carbohydrates. Carbohydrates are hydrophilic organic molecules. So hydrophilic meaning what? We cannot be going this far and we don't know the difference between hydrophobic and hydrophilic. Yeah, water-loving, meaning dissolving in water. And hydrophobic meaning never dissolving in water. Okay. We have this formula, which is CH2O. CH2O. So CH2O times N. So that will show you that all the carbohydrates must have this central formula. Carbon and H2O next to it. Carbon and H2O next to it. Carbon and H2O next to it times N. So every carbohydrate you will see, it will be a sample of this. So glucose is actually 6. Now we put 6 instead of where the N is. Okay, so now let's solve the formula. 6 times 1, that's 6. 6 times 2, that's 12. 6 times 6 times C, that's 6. So glucose looks like that. 6 carbons, 12 hydrogens, and 6 oxygens. You see, right here, glucose N is 6. So the formula is C6H12O6. If it is not like that, then it is not carbohydrate. It is not sugar. It must be CH2ON. So on the exam, CH2ON, what is that a formula of? Peptide bond, proteins, fatty acids, or carbohydrates? Carbohydrates, very good. So there is a ratio of 2 to 1 hydrogen to oxygen, and the names of the carbohydrates often built from the root saccharine and the suffix "ose", both meaning sugar and sweet. Monosaccharides, we talked about those. Monosaccharides are the simplest carbohydrates. They are monomers of larger carbohydrates. So three important monomers are glucose, galactose, and fructose. Glucose, galactose, and fructose. So they are produced by digestion of more complex carbohydrates. They are produced by the digestion. So we know glucose, galactose, and fructose are made by digesting these disaccharides. So ribose and deoxyribose are also monomers. These are parts of RNA and DNA respectively. So here we go. That's glucose, that's fructose, that's galactose. They look like what for us? What do they look like? Isomers. So that's why it's so easy if I have glucose to shuttle it into the glycolytic pathway. But if I have fructose, what do? I convert it into glucose. That's easy because it's a monomer. So I have these interconverting enzymes that convert for us fructose and galactose into glucose. And then we shuttle glucose into the glycolytic pathway. Disaccharides are sugars made of two covalently bonded monosaccharides. Sucrose, that's table sugar. I said that. That's glucose plus fructose. Lactose, that's milk sugar. That's glucose plus what? Galactose. Maltose, oatmeal. I said oatmeal. Now they said grain products. Okay, grain products. That's glucose and glucose. That's the easiest one to know. What are the monomers that make up maltose? So oligosaccharides are short chains of 3 or more monosaccharides. So you see sucrose, you see that sucrose are 2 glucoses. We cleave it in the bottom. We cleave it with hydrolysis or dehydration. Sorry. We cleave these 2 glucoses that make up sucrose with what? Dehydration or with hydrolysis. Hydrolysis. Excellent. Lactose, you see, that's glucose, that's galactose. We break them apart with dehydration or hydrolysis. Then we stop there, or we take the yellow galactose and do what to it? We interconvert it, we change it into glucose. Polysaccharides are long chains of monosaccharides. So there are 3 important polysaccharides. We talked about glycogen, and that is when you have a belt of a lot of glucoses, like a bundle. But a bundle, you know how they used to draw the bundle like that? Now you have a bundle of, not of sticks, but a bundle of glucoses. So that's glycogen. Starch is energy storage in plants and is digestible in humans. So that is the form of glycogen, but it is not in humans, it's in plants, but we can digest it. Then there is cellulose, which is a structural molecule in plants, but it is indigestible. But it acts like a flush. It removes all of the bad stuff in our digestive system. So you see glycogen is like a bundle of glucoses. You see how they are? These are glucose. They're all connected like a chain. They're all connected together. So carbohydrates are quickly mobilized. They quickly mobilize source of energy, and they're all digested carbohydrates converted to glucose and oxidized to make ATP. and they're often conjugated, bound to lipids and proteins and proteoglycans. These are macromolecules that are more carbohydrate than protein. And a moiety is each component of a conjugated macromolecule. Now, we're going to move on to lipids. We finished with sugars. We're going to move on to lipids. Lipids are hydrophobic organic molecules with a high ratio of hydrogen to oxygen. And there are 5 primary types of lipids in the human body: fatty acids, triglycerides, phospholipids, eicosanoids, and steroids. So the types of lipids are fatty acids, and we have essential fatty acids that must be obtained from the food. And there are also fatty acids that are classified as saturated or unsaturated fatty acids. So saturated fatty acids are carbon atoms that are linked by single covalent bonds. And there are unsaturated fatty acids. Saturated means they are saturated with hydrogen. Unsaturated means they do not have hydrogen. Triglycerides, these are long fatty acids that are linked to glycerol. Remember, we drew the backbone of the fat. It looks like this: carbon, carbon, carbon with an OH, OH, OH. That's glycerol. That's glycerol. Then you have the fatty acid. It has an H here, has an H here, and a carbon here, and a long chain, carbon H, and a long chain, carbonyl H, and a long chain. You can see that you can remove H2O, gone. H2O, gone. H2O, gone. That is dehydration. You join them together. When you want to digest, you do hydrolysis, you separate them, you set them apart. You see, that's glycerol. You see glycerol with the 3 carbon molecules, and the 3 of them have, you see, that's OH, and that's and these are the fatty acids. You see how the fatty acids look like? That's their carbonyl group and the long chain with OH, 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 and then they all are going to be joined with glycerol. You see how they are joined? That's how they are joined with glycerol. That's how they are joined with glycerol, and that's how they separate. Okay, that's the structure of glycerol, and that's the structure of fatty acids, and they join together as such. A trans fat is a triglyceride with 1 or more trans fatty acids. Trans fatty acids. So the trans fatty acids are 2 covalent carbon single bonds in the opposite direction. Trans means across from, on each side of the carbon double bond. So the carbon chains are straighter than the cis fatty acids. So all of that, that's not important for us to know. This is just general information. So what's the difference between a trans and a cis fatty acid? The trans has the fatty acids in opposite direction. So you see here, that's the double bond. You see the red? There's a double bond. In the double bond, you have one region pointing up and one region pointing down. That's a trans fatty acid, and that is so one region pointing up and one region pointing down. So when they are separate on different sides, you see that's how the trans fatty acid, because one is up, one is down. But the cis is when they both pointing in the same direction. Do you see the red? You see in cis fatty acid, both of them are pointing upward or pointing downward. You see now? You see that cis, both are pointing in the same direction. Trans, one is pointing down, the other one is pointing up. Is that clear? Lipids. Types of lipids. There are phospholipids, which are similar to triglycerides, but one fatty acid is replaced by a phosphate group. And phospholipids are amphipathic, and that's what we use for the cell membrane. So there is a region in the cell that is facing outside, and that is the head. So they have a hydrophilic head. They have a hydrophilic head. They have a hydrophilic head. They have a hydrophilic head and a hydrophobic tail. So the tail is in the middle and the head is outside. The head. So the head is facing the water. So the hydrophilic head, it loves the water. That's hydrophilic head. That's hydrophilic head. In the core is the tail. That's hydrophobic tail. It's a hydrophobic tail. So the tail is in the middle. That's made of lipids. That's hydrophobic. And the hydrophilic head is outside, pointing to the water region. Okay, phosphate group is linked to other functional groups, and that's the structural foundation for the cell membrane. So that shows us lecithin, which is representative phospholipid. Here, do you see the head? So the hydrophilic head is composed of, you see nitrogen, that's charge. That's hydrophilic, that's charge. In the middle, you have a phosphate group. This phosphate group is what's holding the moiety. And then you have a glycerol, and these are the fatty acids. You see how the phospholipid looks like. So it's not just phosphate and glycerol. There is a nitrogen-containing group known as the choline. The choline. There's a choline at the top, phosphate group in the middle, glycerol, and then the fatty acid. That part is normal. We know that. Glycerol with fatty acids. That's the lipid. We know that one. We know phosphate, this one. The only thing special that we now discovered is the choline. That is at the topmost. So lipids. There are different types of lipids like eicosanoids. Eicosanoids are 20 carbon compounds derived from a fatty acid called arachidonic acid. These are hormone-like chemical signals between the cells. And that includes prostaglandins. So prostaglandins function in inflammation, blood clotting, hormone action, labor contractions, and blood vessel diameter. So prostaglandins are a part of the eicosanoids. And we're going to learn later on that prostaglandins mediate inflammation. Inflammation has cardinal signs, multiple signs of inflammation. You have redness, you have heat, you have swelling, you have pain, you have loss of function in the tissue that is inflamed, and the pain is mediated by the irritation of the nerve endings that is caused by prostaglandins. Yes?
I have a question.
Yeah.
So when you do like D-dimer blood work, does that have to do with this and the inflammation?
D-dimer. The D-dimer is a form of a protein that is secreted during times of inflammation. So it is not identical to, it is not actually a form of prostaglandin, but it is a form that is secreted during times of damage, of tissue damage. So it does reflect that there is inflammation, but inflammation is not only mediated by prostaglandin. And so prostaglandins mediate one aspect of the 5. So inflammation has 5 aspects. Inflammation has redness, red. That's why all the blood is going to the site of the inflammation. That's why it's so red over there. Red, heat, temperature is so hot because all the molecules are going there. Redness, heat, pain due to the irritation of the nerve endings, redness, heat, pain, swelling. So there's a lot of water because blood is running to that region, and there is a loss of function because the inflamed tissue cannot do its function properly. So the D-dimer is reflecting that there is necrosis nearby, that there is damage nearby, but it is not a form of prostaglandins. It's a separate molecule that does help initiate the inflammatory response, but it is not so the inflammation is, like I said, it's 5 multiple things. So you have multiple proteins working together. Cytokines increasing blood vessel diameter, so more blood can rush to the site of the pain, and so on. Okay? Does that answer your question?
Yeah, it's not this.
Okay.
It's not this.
Yeah, it's not this. Yeah. Okay. Any other questions? So eicosanoids have how many number carbons? 20. Very good. Okay, steroids. These are lipids with 17 carbon atoms, and they're made of cholesterol. So cholesterol is the parent steroid from which other steroids are synthesized. So later on, when we learn about the hormones next semester, when we learn about cortisol, when we learn about aldosterone, when we learn about testosterone, progesterone, all of these hormones that are steroid hormones, they are definitely made from Marlon. Cholesterol. They're made from cholesterol. He's the parent. The cholesterol is the big mom who gives us all of these steroid hormones. So when we see steroid, it is made from cholesterol. Very good. Cholesterol. I may be losing my words. Cholesterol. So if you have pancake, it's made from flour. If you have bread, it's made from flour. So the parent is the ingredient that makes up all of them. Here, steroid, immediately think of what? Cholesterol, because cholesterol is the one who makes them. So that's cholesterol. Cholesterol has 4 rings. That's the most distinctive thing about cholesterol. You see right here, 17 carbon in 4 rings. So don't go around counting the 17 carbons. If a molecule has 4 rings like that attached together, 17 carbon with 4 rings, that's cholesterol. How many rings does cholesterol have? 4. Good. There's good cholesterol and bad cholesterol. Good cholesterol is HDL, high density lipoprotein, and bad cholesterol is LDL. That's bad cholesterol, LDL. There is only one kind of cholesterol, good and bad. Cholesterol refer to droplets in the lipoprotein in the blood. So now this cholesterol cannot be carried around in the blood in the form of cholesterol because cholesterol is hydrophobic. So we need a carrier. So it is carried by a lipoprotein. If the cholesterol is more than the protein, that's LDL. That's low density lipoprotein. That's bad cholesterol. But if the cholesterol is a little bit while the protein is more, that's a little bit better. And that's high density lipoprotein. So good cholesterol. You see, lower ratio of lipid to protein. That's what we want. We want a lower ratio. We don't want a lot of lipids. And that will prevent cardiovascular disease. While LDL, low density lipoprotein, bad cholesterol, has a high ratio of lipid. That's why it makes atherosclerosis, the hardening of the blood vessels that nourish the heart, and there's a lot of cholesterol. It contributes to the cardiovascular disease. Okay, a protein is a polymer of amino acids. So protein has multiple amino acids connected together. So amino acids have a central carbon with 3 attachments. The amino acid has a central carbon with 3 attachments. There's an amino group, NH2. We talked about that. That's not a secret anymore. We said that the alpha carbon, C alpha, has an H, has an amino terminal, has a carboxylic terminal, and an R group. Just because you got it doesn't mean everybody did. So I have to make sure everybody gets it. Okay? So did everybody get that the protein is made up from amino acids and each amino acid has 4 terminals and each terminal and 3 of the 4 terminals are shared by all the other amino acids, and that is the hydrogen, the carboxylic terminal, the nitrogen, and there is one side that is the R group. That's what makes them different. Are we clear? Yeah. Oh God.
It's a lot of slides.
Yeah. What can I do? He did one slide less. Fire him.
Okay.
I want you to learn. I want you to learn. I want you to learn. Okay, what do we see here? The amino acids and the peptides. So, what kind of amino acid is that? That's so that's the R group. You see the one in purple, that's the R. Everybody else is not the R. This one right here is the amino terminal. This one is the carboxylic terminal. And that's the H. You see all of them have it. Look at methionine. Methionine has NH2, cysteine has NH2, arginine has NH2, tyrosine has NH2. Then the carboxylic terminal is found in methionine, carboxylic terminal in cysteine, carboxylic terminal in tyrosine. So they all have them. Hydrogen, they all have them. It's the purple group that is what? The R group. Thank God, you got it. Yes.
So you said amino, carboxyl,
Hydrogen. Yeah. Hydrogen. Okay. All right. Proteins. Protein, a peptide, is composed of two or more amino acids joined by a peptide bond. We said that. we said the peptide bond. And a peptide bond is formed by hydrolysis or by dehydration. Dehydration, because we remove the H and the OH to make a peptide bond. Okay, dipeptide has 2 amino acids. Tripeptide, 3 amino acids. Oligopeptide, 10 to 15 amino acids. And the polypeptide is longer than 15 amino acids. So these are the amino acids. You were saying earlier you were having problem understanding the difference between hydrolysis and dehydration. So if you look in this slide now, you see that that's amino acid number 1, and that's amino acid number 2, and that's the H of the amino terminal. So the amino terminal gives 1H, and the carboxylic terminal gives OH. What does that look like? H2O. So we remove the water. Is this dehydration or is this hydrolysis? Dehydration. Water left. They are both connected. That's a peptide bond. That's a peptide bond. So the protein structure, we said earlier, the hydrogen bonds bond together. The hydrogen bonds together. Together, the hydrogen bonds cause the conformational change. So we go from amino acid primary sequence to a secondary sequence by hydrogen bond. They flip on the top of the head of each other and they make that bonding. But if you put temperature, they're going to separate. If you put temperature, the temperature is going to denature. It's going to denature the bonding. So extreme conformational change that destroys the function. So that's why in sickle cell anemia, we denature hemoglobin due to the deformation of the red blood cell. Okay, you see we have a primary structure of proteins. The primary structure is amino acid with protein molecules, and the primary structure is encoded by the genes. We said that in the central dogma of biology. We said in the central dogma of biology, we go from DNA nucleotides into mRNA nucleotides by transcription. Then we go from mRNA nucleotides into protein amino acids. So that chain that we get initially, that's the primary sequence of amino acids. The secondary sequence is when the primary sequence folds up on the top of itself through hydrogen bonding, van der Waals interactions. And the most common type of the secondary protein are your alpha helix. They are spring-like shape, and they are the beta pleated sheets. So usually they ask about this one. So an alpha helix, a beta pleated sheet, is an example of a primary structure, secondary structure, tertiary structure, or quaternary structure. Secondary structure. Yeah, this one is high yield. Knowing that alpha helix and beta helix conformations, they are secondary forms of protein. Then the quaternary structure, these are associations of two or more polypeptide chains due to ionic bonds and hydrophobic and hydrophilic interactions. And that occurs in some proteins. So an example would be hemoglobin. That's a perfect example. A perfect example of a functional protein, protein that's structural protein or functional protein, that is hemoglobin. So hemoglobin is a primary, secondary, tertiary, or quaternary. Quaternary. So there are four levels of protein structure. There's the primary structure, and you see these figures are really nice because these little beads, yellow, purple, purple, yellow, they represent amino acid. So these amino acids connected together, that would give us that would give us a primary structure. So that's a sequence of amino acids joined together by peptide bonds. Then we have a secondary structure. A secondary structure is an alpha helix or a beta pleated sheet formed by hydrogen bonding. Okay? That's a primary structure. That's alpha secondary and beta pleated sheet secondary. Tertiary structure is a single polypeptide chain folded and coiled by interactions among the R groups and between R groups and surrounding water. So the tertiary structure happens when there is interaction between the R groups and water. Quaternary structure, that's when you have your functional or structural protein. And quaternary structure is associations of two or more polypeptide chains with each other. So conjugated proteins contain a non-amino acid known as a prosthetic group, like the iron in hemoglobin. Exactly, right here. So an example would be hemoglobin contains 4 complex iron-containing rings known as heme. So we don't just have proteins. Sometimes we have a moiety. We have an extra group known as the heme group. Okay. Proteins have more diverse functions than other macromolecules. So we said that proteins are structural proteins like keratin, and that is a tough structural protein for the hair, the nails. And then there is collagen. Collagen is found in the deeper layers of the skin, the bones, the cartilage, and the teeth. So that's a structural protein. There are proteins that are functional. They have different functions, like communicating. That's not structural. That's not keratin. That's not collagen. So they communicate like neurotransmitters. So we have catecholamine hormones. So these are hormones that are like proteins. They do the actions of communication. Some hormones and other signaling molecules are proteins. So signaling molecules that exert their effect by reversibly binding to a deeper molecule are called ligands. And the receptors to which the signaling molecule bind are also known as proteins. Proteins are also important because they act as protein channels. So these protein channels allow hydrophobic substances to diffuse across the membrane. And the carriers help the solutes cross the cell membrane via active or passive transport. So carriers, they carry glucose from an area of a high glucose concentration to an area of a lower glucose concentration. And then we have channels that allow the passage and the diffusion of molecules from an area of a high concentration to an area of a low concentration. Then we have catalysis. Catalysis, we talked about that. How does the enzyme make the reaction go faster? By decreasing the how does the enzyme make the reaction go faster? Excellent. It lowers the activation energy that needs to be surmounted in order for the reaction to go to completion. Okay. They're also important for recognition and protection. So glycoproteins are important for immune recognition, antibodies, and proteins. Okay. Movement. So the molecular motors, they are motor proteins and are molecules with the ability to change shape repeatedly. We have seen that in the lipoproteins carrying the lipids. Cell adhesion is when proteins bind the cells together. Okay, we finish at 120. We have 20 more slides. Do you want to break now or you want to go? Okay. So the enzymes. Enzymes and metabolism. So enzymes, we said, are proteins that function as biological catalysts. So some are known as ribozymes. Ribozymes are composed of RNA and found in the ribosome, but they perform the function of being an enzyme for the RNA. Enzymes act on one or more substrates. So they speed up the chemical reaction by lowering the activation energy, the energy needed to get a reaction started. And they permit the reactions to occur rapidly at body temperature. So the enzyme naming convention, we have names for the enzymes such as that end up with ACE, We have examples like amylase. So amylase catalyzes the hydrolysis of amylose. So we talked about that in digestion. So we digest by breaking down the actual chunks of food that we take, or we can break down the food through so first we break it down mechanically, then we use amylase, an enzyme that breaks down the sugar using the chemical digestion. So now we break down from polymer of sugar into disaccharides, into monosaccharides. So do you see the activation energy barrier? So the enzyme lowers the activation energy barrier, and now we are able to proceed faster. Okay, enzyme action. So the substrate binds to the pocket on the enzyme known as the active site, and there's a formation of enzyme substrate complex, and there's an enzyme substrate specificity. It's like a lock and a key. So an enzyme cannot bind a reactant that is not part of the reaction. So we have multiple reactions, and for each reaction, we have its own unique, separate enzyme. That enzyme has like a lock and a key formation. It binds the enzyme binds the reactant and then propagates the reaction by lowering the activation energy barrier and making the reaction go faster. So the substrate sucrose is hydrolyzed by sucrase into a reaction product known as glucose and fructose. So who breaks down maltose? It's maltase. Who breaks down lactose? It's lactase. Who breaks down sucrose? It's sucrase. So the enzyme who mediates that breakage ends with a ase. Okay. So enzyme releases the reaction product. So the enzyme is unchanged and can repeat the product. So it does not get consumed in the reaction. So the example is the substrate sucrose is hydrolyzed by sucrase into the reaction product, glucose and fructose. So that's what we have. That's the enzyme. It has an active site. Remember I told you the pictures, the graphs, these are the main points. So the 3 steps of enzymatic reaction are, we have an enzyme, and on the enzyme we have an active site, and the active site binds the substrate. The substrate is our reactant. Then the enzyme substrate complex are now bound together. You have the active site, the substrate is now on the active site, and then water comes in to hydrolyze the bond, and then we break the bond between polymer 1 glucose, and polymer 2 glucose, and that would be maltase separating for us glucose from glucose. Enzyme structure and action. So the temperature, pH, and other factors can change the enzyme shape and function, and they can alter the ability of the enzyme to bind to the substrate. So if you mess up with the temperature, you're going to destroy the enzyme because it's a protein. Remember, the enzyme is a protein. It has primary structure, secondary structure, tertiary structure, and it's working at the level of the quaternary structure. So if you increase the temperature too much, you're going to destroy the bonds and you're going to disintegrate and destroy. You cause a conformational destruction of the enzyme. So the temperature affects the enzyme. The pH, you can't mess up with the pH either, because if you add too much hydrogen proton, that's going to affect the carboxylic terminal. If you add too much hydroxylic ion, that's going to affect the amino terminal. So we're gonna need to have a set pH for each enzyme. Some enzymes work in the stomach where the pH is so low. Some enzymes work in the mouth where the pH is so high. So each enzyme has its own set of temperature and its own set of pH in order to function. That's an important point. That's an important point, that enzymes do not work at the same temperature and at any temperature or at all temperatures. They have their own specific temperature. And if you heat it up too much, you're going to destroy it. And the enzymes also need specific temperature and they have their own specific pH. You mess up temperature, mess up pH, you're going to destroy the enzyme. The reaction is not going to happen. Is that point clear?
Yeah.
Temperature optimum for human enzymes is usually near the body temperature. So most of like that's what I was saying. Pepsin works the best at pH 2, so low in the stomach, while salivary amylase works best at pH 7 when the pH is so high. Very basic. But temperature, they work at 37. You heat it up more than that, then you're going to destroy it. You're going to destroy the enzyme. Enzymes have cofactors, that accessories. They are accessory and auxiliary. They help the function of the enzyme. So many human enzymes require a non-protein partner called a cofactor. So cofactors may be inorganic or organic. So inorganic cofactors, example of that would be iron. Like for example, iron with hemoglobin. That's a cofactor. It helps hemoglobin absorb oxygen. And you have copper, zinc, magnesium, calcium. That's all inorganic because it has no carbon. And some of these work by inhibiting the enzyme, triggering a conformational change that activates the active site. Organic factors are now called, they're not called cofactors, they are called coenzymes. And they are often derived from the vitamins. An example of that would be the NAD+, nicotinamide. It is derived from niacin and acts as an electron shuttle between the metabolic pathways, glycolysis, and aerobic respiration. So you see, that's aerobic respiration, converting pyruvate into carbon dioxide and water, and that consumes energy. If the reaction points out, that consumes energy. And going from glucose to pyruvate liberates electron that will be accepted by the NAD+. And then the NAD+ will take the electron from glycolysis and hand it over to the aerobic respiration. So you see how it's working. It takes from one reaction, giving it to the other reaction. So a metabolic pathway is a chain of reactions, each catalyzed by a different enzymes. So a simple metabolic pathway may be symbolized as follows. So going from A to B to C to D. So A is the initial reactants. B and C are intermediate. And then finally you have D in the end. That is the end product. So the Greek letters, alpha, beta, and gamma above the reaction represent the arrows, represent the enzyme that catalyzes each step. So A is the substrate for enzyme alpha. So alpha gets you from A to B, and beta gets you from B to C, and C to D is catalyzed by the enzyme gamma. So D is my product, A is my reactant, B and C are my intermediates. So the metabolic pathways are turned on or off by altering the enzyme activity. So this may be achieved on many ways, including the binding or dissociation of cofactors. The end product inhibits an enzyme at an earlier step, and the pathway is turned on when an end product is in demand. So that's ATP. So nucleotides are organic compounds with three components. They have a nitrogenous base. In the case of ATP, it's adenine. They must have a sugar, a ribose sugar, and one or more phosphate. Like ATP has an adenine nitrogenous group, and it has ribose sugar and three phosphate groups. That's ATP right here. That's ribose sugar, that's adenine, and these are the three phosphate groups. If I need energy, I cut one of them and I liberate energy, exergonic, and I keep the rest for ADP. If I need more energy, I break the ADP into AMP, and now I have adenine monophosphate. So the more phosphate I break, the more energy I get. So ATP can give me 3 mole worth of energy. The more I cleave, the more I get. So adenine triphosphate is the body's most important energy transfer molecule, stores energy gained from exergonic reactions. It releases it with seconds for physiological work, and it holds energy in covalent bonds between the phosphates. So second and third phosphate groups have high energy bonds, and most energy transfers to and from ATP involve adding or removing the third phosphate group. The hydrolysis of ATP is catalyzed by adenine triphosphate, known as adenosine triphosphatases. So if we want to break down ATP, we want to break down ATP into ADP. So ATP, it has adenine triphosphate. Adenine triphosphate. I need energy, I break it. Now I end up with adenine diphosphate. So now 2-phosphate. Lastly, if I break that down, I end up with adenine monophosphate, adenine 1-phosphate. So the more I break, the more energy, exergonic energy I get. The enzyme that breaks these bonds is known as ATPase because it breaks down the ATP. It breaks the third high energy phosphate bond of the ATP to produce adenine diphosphate, inorganic phosphate, and energy. ATP is broken down, as you see, to adenine diphosphate plus the one that I broke. Then I get energy. It releases 7.3 kilocalories of energy per mole. And the phosphorylation is the opposite of breaking down the phosphate. So if I want to go up, I go down with catabolism. But if I want to go up, I want to add more phosphate. That is known as phosphorylation. So I add more phosphate, that's phosphorylation, that makes me go up. And the process of adding a phosphate is mediated by an enzyme called the kinase. That's glucose converted into breaking it down into its building blocks, which releases energy, and that joins for us ADP with phosphate to get ATP, and that is causes muscle contraction and or the ciliary beating or the active transport or synthesis or whatever we need it for. That is how we get our ATP. Okay, and we go through the process known as glycolysis. This is glycolysis that we alluded to. We break down the sugar polymer into disaccharides, then monosaccharides. Then we break down the monosaccharides from glucose to pyruvate to get ATP out. Okay, and this is the process, glycolysis, and then we go after glycolysis, we go to fermentation, and that is aerobic respiration, requiring oxygen, where we use pyruvate and we get ATP out of pyruvate. Then we will get to that in next semester. There is guanosine triphosphate. So we don't only have adenine triphosphate, we have guanosine triphosphate. That's another nucleotide involved in energy transfer, and it donates a phosphate group, just like ATP donates a phosphate group. Then there is cyclic AMP. That is an enzyme. And cyclic AMP, the enzyme is known as adenylyl cyclase. It cyclicizes ATP into cyclic AMP. So it gets adenine monophosphate in a cycle, and it's formed by the removal of the second and third phosphate groups from ATP. So the substrate to make cyclic AMP is actually ATP. So cyclic AMP becomes the second messenger within the cell. That's cyclic AMP. AMP, M mono, 1 phosphate, AMP 1 mono, ADP 2 di, tri ATP triphosphate. Nucleic acids are polymers of nucleotides such as DNA, and DNA contains millions of nucleotides, and that is what constitutes the genes. And RNA is the ribonucleic acid, and it assembles amino acids into in right order to produce the proteins. Goodbye, and I will see you.
Thank you.